Our first speaker that I would like to introduce to you is Dr. Isak Gerber. Isak is a principal researcher here at the CSIR. He has a doctorate in biochemistry from the University of Johannesburg. He is a co-inventor on 10 international patents, author of 10 international peer-reviewed scientific publications, and has 8 full process technology packages and 9 technology demonstrators to his name. For the past 10 years, Isak has been an integral member of the team that developed and patented a novel next generation microsphere technology platform called Resyn at the CSIR. The patented technology is currently commercialized with 15 products in its portfolio by a CSIR spin-out enterprise called Resyn Biosciences Limited. So Isak will be sharing with us the ongoing partnership in the microsphere technology, between the collaboration between Resyn Biosciences and CSIR. Please help me to welcome Isak on the podium.
Thank you very much, Amanda, for that introduction. I think you removed about 2 or 3 slides from my presentation. So thank you everyone for attending this presentation entitled An Ongoing Partnership in Microsphere Technology between the CSIR and one of its spin-out enterprises, Resyn Biosciences. It's not advancing. Okay, so firstly, we've prepared a little bit of a biology lesson just for those not familiar with biology, just to basically see and help understand what the problem is that Resyn Biosciences is trying to solve in the international R&D arena. So the large-scale study of proteins is called proteomics, and an illustration of why we study proteomics is perhaps best shown by this image showing how a caterpillar transitions into a butterfly. So genetically, a caterpillar and a butterfly have the same DNA, but it's at the protein level where the differences are very apparent. So the protein profile undergoes drastic changes and transformation to be able to achieve this. So the protein profile can therefore be seen as a blueprint of what's happening at a given point in the life cycle. Okay. So a biomarker, the definition of a biomarker is a biological molecule that can be used to diagnose a state of disease in an organism. So due to the fairly static nature of the genetic material, there's less information that can be gained when you study the genetic material alone. So we look at the proteins to provide a real-time fingerprint of what's happening in the biology. And as I mentioned previously, proteins can undergo a myriad of changes in function, and this complexity alone makes the protein profile very difficult to analyze. And this is where the technology that Resyn developed is coming in to basically dissect these difficult pathways and analyze and fractionate these proteins into different fractions according to their structure, and that's called bioseparation, where you separate the proteins into these fractions. Okay, so what is the Resyn polymer technology? The Resyn polymer technology was developed at CSIR in the enzyme technologies group, and then further in the synthetic biology emerging research area that was part of the CSIR established by the then DSI a few years ago. The technology has several benefits over traditional microsphere or bead or polymer-based technologies, in that as opposed to these conventional technologies, the Resyn technology is comprised of a loosely linked polymer network. If you look at the images there, you'll see it's more like a ball of string as opposed to a solid kind of bead. So what this allows us to do is bind biological or chemical molecules throughout the volume of the microsphere. And this has several advantages. So this gives us superior capacity and functional group density, meaning that all along those strands of the polymer, we can attach biological or chemical molecules, thereby increasing the payload of each microsphere and improving quality of results and improving sensitivity. So this technology was patented, as Amanda said, and it was then commercialized and derivatized into a range of products as part of a thematic bead project at the CSIR a few years ago. So the company Resyn Biosciences was founded in 2012 as a CSIR spin-off and started trading towards the end of 2013. This image shows the current product portfolio of the company. There's about 22 different products in areas of proteomic sample preparation, also some core chemistry particles, and in affinity capture, where people would want to use the microspheres to specifically enrich a particular biological molecule. So the spin-off Resyn Biosciences targeted the life sciences research and development applications with its range of reagents. This type of company is also referred to as a tools company. So it assists in penetrating the market, gaining market traction, and also generating revenue while you are developing these big high-end diagnostic and clinical screening applications. The reason why we decided to target the life sciences R&D applications instead of going straight to the big pharma or biotechs is that the development work starts at the R&D phase. So you need to get into these big companies, biotech, pharma, academia, at the R&D phase. It'll make it a lot easier for your products to then get incorporated into final workflows. You can't just come with an unknown product and try and force it into a current workflow. So that's why we took that approach. So the Resyn company's logo is simpler, better, faster. If that looks familiar, it's because it is. It's Standard Bank's slogan. And Resyn Biosciences obtained full permission from the director of marketing from Standard Bank to use this, because this slogan encompasses exactly what Resyn Biosciences is about. It's not well known in the international community, so it works really well for us to explain very quickly what the company does. In terms of simpler, we try and simplify very complex workflows by scaling up and integrating the workflows into a seamless workflow stream. Then better, we always, when we develop the technology from the beginning, we strive to get products that are better than what was currently on the market. We didn't want another Me Too product. And actually, this technology won a prestigious award at the Society for Lab Automation and Screening in Washington, D.C. in the US as the best new product. So we were very proud of that. And then also faster. Researchers want faster workflows because time is money. So for magnetic beads, we've engineered it. It's magnetic and it's automatable and high throughput. You can see in a little insert video there, it shows a magnetic plate that basically is placed on a magnetic pin, and you can see how quickly the beads clear onto the magnet, and then either manually or robotically, the liquid can be removed and analyzed. So the positioning of Resyn Biosciences in the target market is that we enable the integration of a routine set of experimental procedures or workflows in proteomics to create high-throughput sample preparation workflows prior to sample analysis. Biological samples, by their nature, are really complex. There's a wide dynamic range of proteins or molecules to be analyzed. It's very labor-intensive, and oftentimes the very interesting molecules, for example, in a cancer sample, are the very low-abundant molecules. So the traditional workflows, like I explained, are very laborious and time-consuming. So what we've done is we've basically streamlined and made the workflows more efficient by having products in all the areas of sample preparation. We've got products in the capturing of the sample, the cleanup to remove contaminants, the digestion to make the fractions smaller, also the fractionation and enrichment and cleanup before going to mass spec. So we've got these modular workflows that can be assembled like Lego blocks to form one seamless workflow, and it can be fully automated. So to realize the integration of its technology, Resyn Bio follows a global partnering strategy. I'll explain later as well some of the partnering that we're doing. As an example, Resyn Biosciences partnered with the Olsen Group at the proteomics research in Denmark. This R&D phase resulted in an automated solution to isolate a specific class of biomolecules called phosphate groups, which are very important in cancer signaling pathways, amongst others. So this resulted in an international publication, which has been adopted by a company, Diagnostic, that is used to screen a database of samples called Indivitype. It's a sample repository held by a company, Indivimed. So this publication will also allow other research laboratories to implement this workflow process and to apply it to biomarker discovery and protein profiling. So that's one example where, since we've partnered with an academic institute, it led to a high-impact publication that then got implemented into a real-world scenario workflow. So this is Resyn Biosciences' global footprint of customer and distribution partners. You can see on the map there, most of the customers are located in Europe and across the USA, but we're also now accessing a lot of other emerging markets with the technology. And we've got distributors in all these areas, and that just makes the products more readily accessible to clients and improves the ease of doing business. We've got our local distributor, Separations, which also sells the products to the local African and sub-Saharan African market. A key part of the marketing of Resyn Bioscience is to sponsor scientific conferences and trade shows. And Resyn also sponsors the CSIR scientists to go to these trade shows and exhibit their work. So this allows the proteomics core facility to stay up to speed on the latest developments in their field, and also allows them to form networks and international collaborations and partnerships. We're also very happy to announce a new event on the international proteomics circuit called the Ubuntu Summer School. So this is an inaugural event that will be held in Stellenbosch early next year. This summer school will host 80 local and international students with the aim of transferring knowledge from the leading researchers who will facilitate this event. So for more information, please visit the Ubuntu Summer School webpage. Next, I'll show an example of where the CSIR and the company Resyn Biosciences are working together on sample preparation for new diagnostic technologies. So the challenge with the current diagnosis of acute kidney injury is that the current state of diagnosis by clinicians, a patient already has severe kidney injury with usually less than 50% of kidney function. And it requires extensive and expensive treatment such as blood dialysis. This estimated financial burden of AKI is about 100 billion euros in Europe alone. So it's big business. So in consultation with the clinicians here, there's a strong pull towards discovering biomarkers for earlier detection of AKI. AKI is often a side effect as well of other treatments like antiretrovirals, so it makes it suitable as a companion diagnostic as well. Urine is a great source of potential protein biomarkers, but it's quite a very difficult sample to analyze since most technologies are relatively low throughput and not sufficiently robust to provide high quality sample preparation. So using the Resyn technology, the scientists at CSIR have developed automated sample processing and analysis workflows to basically extract differences in protein profiles between healthy individuals and individuals with AKI. And we are actually proud to say that we've discovered some significantly good biomarkers that can be used for early onset detection of AKI, and the team is now looking for a partner to translate this into a diagnostic assay. Another example of where Resyn and CSIR are collaborating together is to develop robust workflows to detect biomarkers in high-throughput proteomic screening for cancer samples. So this is done in collaboration with clinical partners in South Africa. The benefit of having the proteomics discovery platform in South Africa is the scarcity of the demographically diverse protein samples. So these types of studies primarily take place in Europe and the US, and there's a bias towards data from these ancestries. So the samples in South Africa provide a rich source of data for the discovery of mechanisms behind the cause of prevalent cancers, and it provides a wealth of information for designing new targeted therapies. So it's applicable to our local genetic population. The CSIR has established relationships with local clinicians to analyze the samples, initially focusing on triple-negative breast cancer and colon cancer cells. So similar to AKI, the aim will be to translate this discovery of new biomarkers into a diagnostic technology for the early detection of these cancers, which will improve patient prognosis. So then, lastly, I would like to share a slide on hard lessons learned through this commercialization process. I've been fortunate enough to be part of the development team with this technology from the very early beginning R&D phase through the patent, through translating the R&D to commercial products and then the commercial process. So I've had unique insights in this whole process, and we've learned a lot of hard lessons along the way. Firstly, you have to get out there. That's very important to understand your target market and the customers. You have to go to these conferences and trade shows. Resyn achieved this through participation in these international conferences, because one of the major benefits is that you get to understand the pain points of your customers, not what you think it is. You can actually interact with them and understand the pain points and also derive solutions. And interestingly, a few of our products that are now sold commercially have been developed as a result of scientists interacting with us at these conferences saying, I've got a unique problem, can you develop a solution? So we've developed these solutions because we know how to basically derivatize the tech, and then it got turned into a commercial product afterwards. Secondly, South Africa may not be the right market for your product, the test market, because life sciences customers are very skeptical, we found, at least in our area. And surprisingly, the most skeptical are the local guys. They were very uneasy to adopt or try new technologies. While when we went overseas, they're like, oh, South Africa, let's see what you guys have. We'll give it a shot. And they tried it, and a lot of good things came from it. So that was one lesson that we learned. We thought we first needed to get the local market sorted before we can go international, but that wasn't the case for us. And then MVP, or minimal viable product, does not necessarily work in biotech. That's because biotech customers are used to high-end reagents. So if you go there with an inferior product or a me-too product, that's basically the same as anything else. They might test it and you'll never hear back from them again. So you have to go there making sure that your product is superior in all aspects. To build a biotech economy in SA, startups should target international markets. This is very important because it brings revenue and trade into South Africa, and it also exposes you to a lot of potential partnerships that you wouldn't have had access to if you only target the local markets. Then very important we found is partner. We can't do everything alone. You need to partner with the right guys, the right thought leaders in their field to basically do these kind of product development applications and diagnostic assays like I've shown with an example with Biognosis and partnering with the guys in the Olsen group. That it makes a huge difference if you partner with the right group. And then also be prepared for a rainy day. Things can and will go wrong, and fixing mistakes can be costly and expensive. So always be prepared for that, and nothing goes smooth as you think. And lastly, and also very importantly, it takes time. Everybody thinks you've got such a good product and you're going to shoot the lights out, but it takes time. We learned the hard way. It took us years to get market adoption. The first sample we sent out to a university in Scotland sat on their shelf for 2 years before they tested it. And we had nobody else to test it. Then one day, all of a sudden, we got a mail saying, this is wonderful stuff. Can we have some more? So you have to wait. You have to wait sometimes for these guys. You send samples, and then it takes time. So, be prepared for that as well. And that's it from me.